And next we have Jordan to give us the topic of inducive gene silencing. Yeah, sorry for that. Thank you.
Good evening, everyone. First of all, thank you for the organization to invite us to allow to show our project, especially Elena for her work. Today, I'm Jonatan Niño Sánchez. I work in a group of Julio Diez Casero. We belong to the I don't remember the name of the department. Is vegetal production and forest resources, I think, right? So today, me and my lab partner, Irene, we are going to explain one of the projects that we have in the lab. And it's about RNA interference, and it's kind of molecular biology. So we decide to split the talk in two parts. So firstly, I'm gonna introduce like a brief introduction about biological, about molecular biology, so sorry. So the title of my talk is Spray-Induced Gene Silencing 6 provides RNA interference effect against fungal infection on plant material. So I would like to explain the concept of spray-induced gene silencing and also RNA interference and put all of you on the same page. So for that, I will start by the beginning. And the beginning is this guy whose name is Francis Crick. He was a genius. I think you know him because with the help of Watson and Rosalind Franklin, discovered the helical structure of the DNA. But also he's important because he was the first person to describe the central dogma of the molecular biology. So I include this picture. This is 1957, and this is the first time that he talked about the dogma of the molecular biology. And in fact, he's saying that DNA makes RNA and RNA makes protein. Let me make some zoom to his blackboard. And what is in the blackboard is that DNA can replicate itself. This process is, for example, in the cell division, right? But also DNA has the information to make RNA, right? And actually messenger RNA. Messenger RNA is single-stranded, while DNA is double-stranded. And RNA has the information to make protein. Protein is really important in the organisms because are the molecules that has a lot of kind of functions. Another molecules have other functions, but all the proteins have different functions. So in our research field, we work with fungal disease. So we know that actually the best or the more efficient to control fungal disease right now is using the fungicides. And fungicides usually target some proteins, right? But we know the fungicides is not good for the environment and also can create new fungal resistance because fungi can easily change DNA to make different proteins to escape the fungicides. So we are looking for new regulations to reduce the protein production. And as I said, this is the central dogma, but this is really incomplete because it has a lot of extensions and regulations inside. So I'm going to focus on one of this, and I'm focused in the role of the microRNAs. In this slide, on the left, you can see in blue the central dogma, DNA that has information for the protein coding makes messenger RNA, and RNA makes protein, right? But in green, you can see another parts of the DNA that has not information for protein coding, but can make RNAs of different types. For example, microRNAs, those ones. MicroRNAs has the feature that they are double-stranded, and double-stranded RNA is really weird to find in our eukaryotic cells. This is why eukaryotic cells have evolved to create an RNA interference machinery to try to destroy these microRNAs. So the eukaryotic cells can chop in small pieces around 20 to 25 nucleotides of double-stranded RNA that we call small interfering RNAs. These small interference RNAs are able to target some messenger RNAs like this, and then another protein recognizes this junction and can cleavage the messenger RNAs. So with this process, at the end, you have no protein. So this process is called post-transcriptional gene silencing, or PTGs. This process came from the defense against virus, because virus, a lot of virus has some double-stranded RNA, and as I said, double-stranded RNA is very weird in eukaryotic cells, so eukaryotic cells can recognize the virus dsRNA and chop this, and to prevent the virus replication. The second concept that I would like to explain is the cross-kingdom RNA interference. Here you can see two cells. The green one is a plant cell, and the blue one is a fungal cell. Just suppose that this fungal cell is pathogenic to the plant cell. So it is known that fungus can produce a small interfering RNA and transfer this small interfering RNA to the plant cell to inhibit the expression of genes related with the defense, the plant defense. At the same time, plant can also produce a small interfering RNAs and transfer these small interfering RNAs to the fungus to inhibit the expression of genes related with violence. So this is why I said that this cross-kingdom RNA interference is a bidirectional process. So in here we can see an opportunity to control the fungal disease, because we can create plants that can specifically express small interfering RNAs against fungus. And this technique is called HIGS, host-induced gene silencing. But the problem of this is that it involves transgenic plants, so we cannot bring this technique to our forests, right? So how can we overcome the transgenic plants? So it's kind of easy. So we can just spray this small interfering RNA on the fungus. So, and this technique is called spray-induced gene silencing, SIGS. Okay. So the first time that SIGS was described was in Caenorhabditis elegans, which is a nematode. You can see in the picture genetically modified Caenorhabditis elegans that is able to express GFP. And when you spray on the nematodes the small interfering RNAs against GFP, you block the expression of the GFP, so under microscope you don't see any fluorescence from the GFP. So it's known that SIGS also works in fungi, plants, insects, and mammals. And this is an example in fungi. actually in Botrytis cinerea, which is a very important pathogen that can infect more than 1,000 plant species. So in here you can see how if you spread with your small interfering RNAs against a couple of genes that are important for botrytis, and then you inoculate with botrytis, you get a reduction of the infection in several plant materials. It has been proved in a lot of fungi, for example, in Fusarium graminearum, in monocots like barley or wheat, and it works. And finally, I just want to say that not all the fungi are good for SIGs. The fungus must have two requirements. The first one is that it has to have a complete RNAi machinery, okay? And the second one is that the fungus needs to able to uptake the environmental double-stranded RNA. And finally, I think Irene is going to talk you more about this experiment, but just briefly, we can label double-stranded RNA with fluorescein, and we can spray on the fungus, and we can see if, under microscope, if these dsRNA are inside the cells or not. So this is a good assay to check if they are if our fungus can uptake or not the RNA. So here there are some advantages about SIGs. I'm out. Okay. And that's everything. Thank you.